Many people use drugs, right? Illicit drug use in the US, and this is in numbers of millions of individuals. So around 25 millions of individuals endorsed using drugs in the past month. Marijuana is the highest actually in terms of use, 20 million of individuals, and cocaine that we study in our lab is used by 1.5 million of individuals in the past month if they are above the age of 12 or older. And cocaine is also rated as the second highest after heroin in terms of the physical harm and dependence that it creates in people who use it. Cocaine is also very harmful in many different ways, and actually it's #1 drug that brings people back to the emergency department visits. And it's more so than marijuana-associated emergency department visits or heroin and other opiates. So it's a very dangerous drug. Avoid using it. However, drug use or abuse or even occasional use, even frequent use, is not drug addiction. And I wanted to talk a little bit about what is drug addiction and how it's separated from drug use and abuse. Drug addiction is also not just bad habits, looking at your iPhone every 1 second. That can be a bad habit. And even drug dependence cannot explain drug addiction, although drug dependence and abuse and all of these things are part of addiction. So only with addiction you have a loss of control over drug intake, despite catastrophic consequences to the individual's life and despite a decrease in pleasure that's derived from the drug. So if you think, for example, about drug dependence and how come it's not part of addiction and it's something that can kind of boggle your mind, think about people who sustained accidents, who go through very major accidents. They go to the hospital, they receive morphine or other type of opiate painkillers, and they can become, of course, very dependent on them. You can see that by the fact that you need to increase the dose to get the same effect, and if you take it away, there will be withdrawal symptoms. And still, it's not addiction. It will become addiction if the person starts using it more than prescribed and more than basically is going beyond the pain killing. And when the drug becomes so pronounced and so central and everything else basically recedes to the background. So other things that are extremely important to the individual, their job, their family, their goals, can wait. Everything can wait, but the need to do the drug and the next dose, that cannot wait because that becomes a survival issue. And to really understand about the phenomenology of addiction, I recommend, those are wonderful books. There are many other types of books, and we can talk about that later. So how do we understand addiction in my lab? We are thinking about the IRISA model of addiction, where we emphasized that impairments in response inhibition and salience attribution underlie those core clinical symptoms of addiction that have to do with craving, severe craving that develops for the drug, binging with some drugs where you have loss of control over the frequency and amount of drug use, very severe withdrawal symptoms that then go back to feed into this chronic use of the drug use and drug self-reinforcement, despite the best intentions not to do it. So we think about it as a chronically relapsing brain disorder, and we think about neuropsychological mechanisms that have to do with self-control and the ability to attribute and assign value and salience in the environment as really integral part to understanding addiction. And we focus in our review of addiction, we focused on the prefrontal cortex and the importance in understanding its role in higher order executive functions as relevant to addiction, kind of expanding the usual view that comes from animal studies beyond subcortical system and striatal type of systems that underlie hedonic responses and habit type of formation. I'll show you results of many studies. All our subjects are cocaine addicted. They're smoking crack cocaine for about 20 years, for about 4 days a week. Mostly are male, mostly African American, and mostly in the mid-40s, with high school education and average intellectual functioning. So they are representing the Northeast urban New York type of area. And we always compare our cocaine-addicted individuals to people who are not addicted to cocaine. We can match them on many of these demographics, the sex, the race, age, education, intellectual functioning, but some variables that are part of the phenomenology of addiction, we study them and we focus on them. For example, other drug use, so nicotine use is very common in drug-addicted individuals, also alcohol, marijuana, other drugs, and depression that is more than minimal. And sometimes we statistically control for it, sometimes we match the groups on those variables, and sometimes we just focus on their importance in understanding addiction. I hope to have enough time to go over 5 major concepts, and not 1 or not 2, but I think that but they're essential to, and it gives you just a kind of bird's eye view and an introduction to this day, I hope. So one is that attention and higher order cognitive function are impaired in drug-addicted individuals, and it is associated with the functioning and the integrity of the prefrontal cortex. Everybody knows about Phineas Gage and what happens to your decision-making when your prefrontal cortex is basically, or the orbitofrontal cortex is blown. I'm assuming the knowledge of that here. How about the Red Baron? So this is another classical neuropsychological case that shows how a lesion to the prefrontal cortex affects not only personality but behavior in such a way that it causes basically extremely disadvantageous behavior that cost a person's life. So the Red Baron was a World War I combat pilot, and it not only was flying combat airplanes, but actually was commandeering a whole fleet of planes. He was widely respected across all lines for his discipline, the aerial strategies and tactics. And similarly to Gage, he sustained a lesion to his prefrontal cortex during one of those combats, but he was able to recover, and to such a high level that very quickly he was back flying an airplane during combat. This requires very high level motor and visual motor type of and other type of functions. But he died 9 months after the accident because he did something that every very disciplined and experienced airplane combat person knows not to do, which was flying in pursuit of an adversary very low and crossing the enemy line. And he was promptly shot down. And this is a classic example of the inability to discontinue a behavior, a fixated behavior that is very disadvantageous to your livelihood, and it basically was killed. So I think that the Red Baron case, that classical neuropsychological case, is a wonderful example for addiction and the inability to discontinue a behavior that is clearly very disadvantageous. So if you test people with this type of lesions with classical neuropsychological tasks, you may not even see any type of impairment, and you have to look at decision-making tasks. There are many of them, so the Iowa gambling task is one of them. I don't know if people are familiar with it. Great, so you have to maximize your gain. You have to be able to let go of immediate gain that is high for longer term, to avoid longer term loss, and you have to find out the bad decks versus the good decks. I'm just going to skip that. I'm assuming people know that. And so those type of behaviors where people are, they need to find those type of good decks over bad decks, and you have to suppress going for something that's immediate reward for longer term avoidance of punishment. People who are healthy controls, they discover the good decks and they avoid the bad decks as they go along. But people with those type of lesions, TBI, and people with addiction, they go back to the bad decks. Maybe this time I can get the right reward. Maybe this time I will not get the punishment. So that has been shown. And when you compare healthy individuals to people with the substance abuse disorders, and that is now a replicable finding that has been shown in many different labs across many different addiction types, and this is results of structural MRI, and it's so quantifying the volume of gray matter with voxel-based morphometry. So basically, you are classifying a voxel based on its intensity and prior probability maps. You're looking at the tissue types, you're looking at gray matter, separating it from white and CSF. And if you compare the groups, you can see a decrease in gray matter in the rostroventromedial PFC, the frontal region of the orbitofrontal cortex in the medial parts, in people who are addicted as compared to control subjects. And the important thing here is that the lower the gray matter, the more the disadvantageous decision making, the more the playing and the choosing the bad decks in the people. So the more you have decreases in this gray matter, the more you have impairments here, the more you're playing bad on those and you're making disadvantageous decisions. So maybe this type of mechanism can explain or can contribute to that decision, that disadvantageous decision of going back again and again to using drugs over time. And we can attribute it maybe to some kind of morphological integrity impairment in the prefrontal cortex in drug-addicted individuals. So we have in our lab, this is work by Nelly Alea Klein, who is the co-chief of our NARC lab. We see similar decrements in the orbitofrontal cortex in cocaine-addicted individuals as compared to controls. And this time we saw correlations with lifetime years of cocaine use. So the lower the gray matter, the longer the lifetime years of cocaine use. I want to remind people that this is a correlation. We cannot attribute any causality. So because maybe another factor predisposes individuals to both decreases in gray matter and more severe and longer drug use patterns. But it does suggest that if we are able to somehow help people discontinue drug use, maybe we can help them preserve the integrity of their prefrontal cortex that they need so badly to make the right decisions. Another point I want to make on this slide is that not only correlations with the orbitofrontal cortex were seen with lifetime cocaine use, but also other regions are crucially important. And this is showing the hippocampus that correlated not only with lifetime years of cocaine use, but also with lifetime years of alcohol use and nicotine use. And the hippocampus is extremely important, as we all know, to our ability to extinguish responses and associations to stimuli that are no longer rewarding. So that ability to discontinue responding to something that used to be rewarding is no longer rewarding, maybe predisposing people to relapse. So what happens? The question is then, we cannot attribute causality, but maybe we can think about this in more mechanistic ways, despite the fact that with cross-sectional imaging studies, again, we cannot attribute causality. But still, let's think about it mechanistically, and this is results by Muhammad Parvaz. He's an assistant professor in our lab at Sinai, answering the question, what happens with abstinence? So these are cocaine-addicted individuals. They were scanned at baseline immediately after treatment as usual, and then 6 months later with abstinence, and mostly abstinence with significantly reduced drug use. And you can see that with abstinence of 6 months, there is recovery of that gray matter measure, again, in the inferior frontal gyrus and the ventromedial prefrontal cortex that I showed you before. And our type of results cannot answer the question of what exactly is the neural mechanism that underlies this recovery. What are those synaptic markers? Is it axonal cone growth? Is it dendritic spine density? Is it glutamate concentration? We cannot answer that directly, but we can answer questions on behavior. And what does it mean, this recovery of the actual morphological integrity of these regions of the prefrontal cortex to behavior? So here is a quantification of behavior on that decision-making task that I showed you, the Iowa gambling task. At baseline, this is the net score. So there, people with cocaine addiction are, again, choosing the bad decks. But at 6 months later, they no longer show differences between with the healthy control subjects. So this is the controls were also scanned at baseline and 6 months later to look for practice effects. And here there is a clear recovery of function, of advantageous decision-making in cocaine-addicted individuals at 6 months later that was correlated with this recovery of the structure in their brain regions. So maybe with recovery, people gain mechanistically, behaviorally, and in their neural networks, in the neural correlates of this behavior, they gain that ability to make the right choices. Or maybe it is a very selected subgroup that was able to abstain for 6 months. And the people who are able to abstain also are able to make those type of decision-making that is good for them and for their abstinence. So beyond decision-making, we see cognitive impairment across different measures that we look at. And I want to make this point and then again bring it back to retention in treatment. So I showed you the Iowa gambling task, but if we measure other cognitive functions, visual memory, verbal knowledge, verbal memory, attention and executive function, and we compare cocaine-addicted individuals or people with alcohol dependence to healthy control subjects, we standardize the mean of the healthy controls to 0 and this is severity of impairment in Z-score, so level of severity of impairment. And you can see that across those different tasks, there is an average severity of impairment that's below 1 standard deviation, the healthy control mean. If you compare it to people with schizophrenia, for example, statistically, the severity of impairment is not as bad. In people with schizophrenia, it's around 1.5 standard deviation below the healthy control mean. And that may explain how addiction is one of the diseases that is still not accepted by everyone, even scientists in the field, as disorder of the brain. I think this is one of the reasons, is that to find the impairment, you have to really look for it. Because statistically significant doesn't mean you can see it clinically. And some of us in this room, in this conference, can be addicted. But you will not see it unless you put us in the right context. Because to express addiction, you have to be in the right context. So that is one point. But despite the fact that it is so mild statistically, it affects people's life. And we can see that based on correlation with retention in treatment. So this is global cognitive functioning, showing that the higher the global cognitive functioning in people with cocaine addiction, the lower the odds of attrition from treatment. So it means that if we can somehow increase cognitive function, if we can improve cognitive function in people with addiction, we can then maintain them in treatment, and we can teach them to use the techniques that we have to help them abstain from drugs. Okay. So that was one point. And now I'm going to talk about the salience. I spoke about cognition, retention, morphological type of integrity, and how addiction is not traumatic brain injury, the importance of the context, and now that type of what happens with the salience, and how attention is grabbed by drug-related cues away from other very salient cues in the environment. So the best demonstration of that, and we have so many, but the best one is actually an event-related potential study where people are just passively looking at images that are pleasant, that are horribly unpleasant, that are neutral, or that are drug-related, while we record their electrical activity from the scalp. So those are basically, and then quantify the event-related potentials, which are positive and negative deflections in the ongoing EEG that is time-locked to a certain event. This time it's just time-locked to looking at those images. And we quantify here the late positive potential. Potential is one component in the ERP. We quantified it at 2 time windows, the early window and then later window. And we always compare the pleasant images, the salient ones, to the neutral ones. And then the unpleasant one also to the neutral, and the cocaine to the neutral. So these are healthy control subjects. We see a robust activation or robust response to these images and to the unpleasant images, and nothing quantifiable statistically to those type of cocaine-related images. And if you're a healthy person and never used this type of drugs, those type of little bags can look like what? Bags of sugar? I don't know. Well, sugar is not that neutral, but it can be. Anyway, there is no response at the late positive potential. But if we look at our cocaine-addicted individuals, the first thing that jumps into our view is the response to those images as compared to the neutral images. So clearly, it's not neutral. And another thing that we see is that for the pleasant images and the unpleasant images, at the second time window, the longer they process and they look at those images, the less is the response. So there is more habituation, more emotional type of dampening to images that are extremely salient, whether they're very pleasant or unpleasant. And this, the late positive potential is a measure, is a bottom-up type of measure. So it shows an automatic type of grabbing of your attention to the drug-related images and some persistence and more type of in-depth processing of these images that remains, but you have a reduction of processing of these other salient images. And again, what happens with 6 months abstinence? Again, it's work led by Mohammed Parvez. And here we show the same results I showed you before, just with a different type of display, comparing the pleasant to the neutral images at baseline in cocaine-addicted individuals only. And you can see this dampening that I showed you before. And 6 months later, with abstinence, you see a recovery at follow-up to the pleasant images as compared to the neutral images. And a very crucial contrast that we always have to think about in drug addiction research is the contrast not only to a neutral stimulus to control for the motor and the visual and the sensory input and output, but the comparison has to be to another salient stimulus to really understand addiction. Because people with addiction, they go for the drug-related cue over cues and stimuli that are really salient and valuable to them. And that's the point in addiction. So we have to make that contrast. And this is the contrast of the cocaine images to the pleasant images at baseline. And you can see this is cocaine more than pleasant. So the late positive potential component is higher for the cocaine images as compared to the pleasant images in the cocaine-addicted individuals at baseline. But with 6 months abstinence, you have the reverse. So suddenly, they do respond to the pleasant images more than to the cocaine images. So there was a flip, and they're able now to respond to just regular, pleasant, salient stimuli in the environment. So there is a recovery of function that may be explaining recovery of hedonic functions. People think about addiction as a high and the pursuit of pursuit of happiness in some way, but it's actually a disorder of, if anything, of pain. And we have to think about that people are using drugs despite very negative consequences and withdrawal, and to alleviate their type of symptoms. So here there is a recovery of response to hedonic stimuli. Okay, so what can we do in addition to abstinence, in addition to waiting, because we are kind of an impatient bunch in my lab. And 6 months abstinence is something that is achievable by some people, but not by most people with addiction. There is a huge relapse. So here we use this for cognitive intervention. And can we do something about it? Can we train people to respond correctly? So here we expose them to drug images, neutral images, or unpleasant images. But this time we use reappraisal techniques. So people either can look at those images, or we ask them to decrease your response to the image. So we really ask them to reinterpret and not just distract or avoid looking at the image. So for example, you can think to yourself, yeah, okay, it's a person smoking crack, but it's a movie. It's not my crack. I cannot touch it. So you have to really reinterpret the image. And then the most important part is that after this type of reappraisal, we measure their spontaneous eye gaze. So the duration of time they will look into those they will look at those images that can be the drug-related, neutral, or unpleasant images, or a black screen for control. We also ask them questions to rate how much they like the image and how much they want it as assessment of craving. Gaze duration is the time spent looking at one cue over the other. It assesses attention bias, and cognitive reappraisal strategies reduce gaze duration to salient stimuli in health. That has been shown before, so we did it in cocaine-addicted individuals. And these are our pilot results. It's not preliminary results. It's not yet published, showing that in healthy controlled subjects, you can see here the drug-related gaze duration relative to neutral images. And this is during the look and during the reappraised conditions, and then the direct contrast. But what's important here is that in the individuals with cocaine use disorder, you see a reduction in gaze duration during reappraisal. So after they reappraise the images, after they're able to somehow reinterpret the situation, we measure the spontaneous gaze duration. This is not instructed. It means that after they reappraise, they're able to generalize spontaneously, and they don't look as long at the cocaine cues. And this type of attention bias, this automatic attention bias, is reduced. Maybe we can use this type of training and reappraisal then outside the lab, and maybe with repeated type of training, they will reduce their immediate automatic responses to drug-related cues, and maybe then that can help them avoid using the drugs, or at least reducing the amount of use. And this has been suggested before by a group in the UK that used the Stroop effect. Basically, you're looking at an image, you're a person who is a heavy drinker, and you're looking at an image of your most favorite drink. It can be a nice bottle of white wine or whiskey or whatever it is. They really tailored the picture to the person's most favorite drink. You're looking at that, but what you have to do is press for the frame that surrounds the image. You have to press for it as fast as you can for the color of it. And people can be trained to do it faster and faster, meaning they're able to ignore that image and what it contains. And the interesting thing is that after training, there is a reduction in alcohol intake by around 10 units a week. So that's 10 drinks less a week, which is a lot. It's significant for the individual's life. And if you ask them, so these people that receive this simple cognitive training in the lab, you ask them, so what happened exactly? How come you reduce your drinking by 10 drinks? And this is in London, where you can buy alcohol, heavy alcohol, in the supermarket. So they said, oh, we just didn't go to the aisle where you can buy the alcohol. So they learned, they generalized that type of cognitive training in behavior to avoiding the context of that is drug-related and that induces them to drug seeking. Okay, so beyond, how am I doing on time? Okay, thanks. Okay, so beyond abstinence, beyond cognitive training, what else can we do? Because we really want to help people with addiction. It's a lethal disorder. People die from it. Young people, not that old people are any less important. Okay, so we can use pharmacology, we can use methylphenidate. And that's what we, or many other types of drugs. But basically, we chose in our lab methylphenidate because it is very similar to cocaine in terms of blocking the dopamine transporters, increasing extracellular dopamine. It increases the salience of food stimuli and math tasks in healthy control subjects. It bolsters cognitive functions by improving signal-to-noise ratio and optimizing the activity of the brain regions that actually support that type of cognitive function and accurate task performance. And if given orally, it decreases risk-taking and improves inhibitory control in psychopathologies that also affect the prefrontal cortical integrity and function, such as frontotemporal dementia, ADHD, TBI, stroke, and I forgot to mention methylphenidate is basically a stimulant we give to children with ADHD. It's Ritalin. So the hypothesis is that methylphenidate will improve brain behavior function in cocaine addiction if we administer it at low oral dose in combination with some lab challenge. And it's not that surprising because interference with binding of a drug, but different pharmacokinetics, so slower ones, is valuable in the management of other drug types of addiction. So for example, replacing heroin with methadone and nicotine, cigarettes with nicotine patches. It's the same concept of replacement therapy. So we did that in the scanner, of course, during imaging, because we like to understand the mechanism underlying any type of behavioral change. And this is a placebo-controlled study where we give either methylphenidate or placebo. People don't know what they're getting. They cannot even guess what it is because it's low dose. And then we can scan them. We can do a resting state scan for functional connectivity. We can, they perform tasks. And then we can do another type of resting state connectivity scan during peak effects of methylphenidate. And we can measure methylphenidate in plasma. And it is the peak effects of methylphenidate reach after 60 minutes, and then they stay there for 2 to 3 hours, basically. So we have a nice long window where we can scan and assess individuals whether on placebo, they're not getting anything, they don't know they're getting a pill, but it's not active, or they're getting methylphenidate. And we have this window where we can scan them while the effect of methylphenidate is at peak. So in addition to scanning them at resting baseline, so they're not doing anything, we also have them perform some tasks. And this is our drug Stroop task in the MRI. And I mentioned the Stroop effect before. I'm not going to show it. I'm just going to walk you through it. So you have to press for color of words. And the words can be either drug-related, red crack, or neutral words. And you respond for their color, basically. So you have to suppress what you read, and you have to just respond fast to the color of the word. And we also give money. So there is an incentive, a reward, and we have different levels of the reward. So basically, it's a very salient task. It has drug use, it has money, it is an engaging task. And we matched our groups, the cocaine-addicted individuals and controls, on performance on this task. We wanted to see, are there differences in how their brain reacts to this task, even if at the behavioral level you cannot see anything. And we indeed see this type of decreases. This is with matched group performance in the dorsal cingulate and the ventromedial PFC I mentioned before. So here is the dorsal cingulate. The activity to this task is decreased in cocaine-addicted individuals as compared to control subjects. This is a region that's essential for cognitive conflict monitoring, resolution, error detection, performance monitoring, and reductions. There are reductions also in the ventromedial PFC in cocaine-addicted individuals as compared to control. And this region has lots of essential functions. Some of them are with emotion regulation, value attribution. We repeated this study in different cohort of subjects, this time with the placebo and methylphenidate. And again, at placebo, you see this type of decreases in the dorsal cingulate in cocaine-addicted individuals as compared to controls. But with methylphenidate, there are no longer decreases. There is a recovery of function in this region that you can also see in the ventromedial PFC here. You have the decreases in activations at placebo, but not with methylphenidate. And this was associated with the recovery of behavioral function. So with methylphenidate, there were also less errors of commission, which is a measure of impulsivity, and there were correlations in the recovery of the brain function and the behavior. So basically, methylphenidate, giving this low oral dose to stimulant addicted individuals, improved their function while performing this very salient task, reducing their impulsivity. What happens to the connectivity between the brain regions? So we can assess with fMRI BOLD the synchronous fluctuations in the signal that is indicative of fluctuations and synchronicity in neuronal activation between the brain regions. So brain regions communicate, and we can quantify it. Between the regions in the ventromedial PFC that I showed you and the dorsal cingulate, the red line is healthy controls. At placebo, cocaine-addicted individuals show decreases of that connectivity. With methylphenidate, there is recovery of that connectivity. So the brain regions start communicating more efficiently with methylphenidate. And we see recovery not only between these prefrontal regions, but also subcortically. So subcortically, between the seed here is in the ventral striatum, the location of the nucleus accumbens, which is crucial to hedonic responses, and the dorsal striatum, the putamen, where we can see at placebo in cocaine-addicted individuals hyperconnectivity. There is too much connectivity between those regions of the subcortex, of the striatum, the ventral and dorsal. And methylphenidate reduces this abnormal connectivity in cocaine-addicted individuals. And that type of hyperconnectivity here at placebo is related to severity of addiction. So the more it is, the more severe the addiction. This is abnormal. And with methylphenidate, we reduce it. So these results actually go back to the concept of spiraling dopamine connections through the midbrain that link the nucleus accumbens, the ventral striatum, with the striatum, the dorsal striatum, implicated in drug seeking. And there is the concept in addiction of a progression that involves the shift in striatal circuits between the ventral to the dorsal circuit. So between the hedonic, in responses to hedonic stimuli, to habit formation as linked to disease severity. So maybe weakening this connection, maybe chronically, not just acutely one time that we did here with one pill, maybe we can help people, again, abstain from drugs, which is our goal. And that has been suggested in animal studies, not yet shown in human studies. Okay, so the last concept I do want to mention, I think because there is a representation also here of the psychology, just general psychological thoughts, concepts, is the concept of insight and self-awareness that have been heavily studied in classical psychiatric and neurological disorders, schizophrenia, neglect with stroke, but not in addiction. So this is past year perceived need for an effort made to receive treatment among people who need to be treated. So this is 20 million of individuals who need but don't receive treatment for drug use or alcohol use. 96% of them feel they don't need treatment. Okay? So almost 100% of people with problems with drug use and alcohol use feel they don't need a treatment. Maybe because addiction is stigmatized, which it is. And some people think about it as denial. Maybe they're in denial, maybe they're lying, which is also, again, concepts that are very much associated with addiction. But we, of course, think that it may reflect this type of avoidance of treatment. It reflects impaired awareness of the severity of disorder, and that we can actually quantify it in the lab. And that, of course, it's associated with some dysfunction at the brain level. So we designed our tasks to assess, how do you assess awareness and insight? So tell me, how aware are you? I'm 100% perfectly aware all the time. We all have our blind spots. And if anybody asks us about them, we have a blind spot. We cannot tell, right? So how do we assess it? Okay. So here, instead of the Iowa gambling task, we still like this deck type of display of cards. And you have to choose at what is it that you're going to look at. It can be deck 3, and then it flips over, and you see this nice, pleasant image, and you can vicariously experience a vacation that becomes very, very pleasant the longer you didn't have a vacation. And then you can sample from the same deck. Or you can say, no, you know what, I want to see what other things are there. And you can sample a different deck, and then here is a crack image. Now, the importance of this type of task is that so each deck contains pictures that belong mostly to a certain emotional category. I showed you the pleasant category, the drug-related category. We also have unpleasant images and neutral images. But we stack them such that people are not sure what it is exactly, because although most of the images in a deck belong to a certain category, we also put some from the other categories, and actually a deck choice begins with images from the other categories. So people are not sure what it is they're choosing, and after 8 choices from the same deck, we flip the whole thing again. And then they have to again find out what it is that they're doing. We also have an explicit version of this task where people just look at 2 side-by-side images, and they have to press for the image they want to look at, basically. So, okay, in the explicit task, we compare people who are cocaine-addicted individuals who come to our lab, and they can be positive or negative for cocaine in urine. All of them are addicted, but some of them used cocaine in the last 3 days, which is the resolution of our urine test. So up to 3 days before the lab, they come to the lab, and some of them are abstinent for longer time, and their urine is negative. So you can see that the choice for cocaine images is highest for the cocaine-addicted individuals who are cocaine urine positive, but it's also high for the abstinent ones, more than the healthy control subjects. And the important thing is that with the implicit task, we see the same type of bias of choice. You choose to look at cocaine images if you just use cocaine the last 3 days, you're a current user, or if you're abstinent more than if you are a healthy control subject, and there is a flip of your choice of the pleasant images. So if you are a healthy control subject, you look at the pleasant images more than if you are a cocaine-addicted individual, whether abstinent or current user. So again, that flip and that bias now in choice, not just passive viewing, but at the choice level. And this is work, a series of studies by Scott Moeller, an assistant professor in the lab who's moving now to Stony Brook University. This type of task at the lab level is important to understanding behavior outside the lab. And here we can quantify this type of choices, again, comparing cocaine to pleasant, to that hedonic choice, and look at drug use as reported in the last 30 days, but also 6 months later. So this is their choice in the lab at baseline, and here is the total drug use 6 months after they perform the task in our lab. And the more the type of cocaine, the pleasant choices in the lab at baseline, the more the drug use 6 months later. So this task can prospectively predict drug use in cocaine-addicted individuals. So it has validity to behavior outside the lab. Okay, but going back to awareness, it's a great task and it has validity and it's reliable, but what does it mean and how do we use it to assess awareness? So its implicit contingencies and its probabilistic nature allowed us to ask this question of what kind of pictures do you think you chose to look at most often? It's a very simple question. And so we can, and people answer it, and then we can compare what people did on the task, their actual choice, to what it is that they said. And that's how we can try to assess those blind spots. Is there a difference between people, what people say and what they do? That's one way. Other people assess it by a difference between what people say and other people say about them. That's another way. But we used a person's data. So healthy controlled subjects, most of them are very reliable reporters about what it is that they chose to look at most often. But in cocaine-addicted individuals, we see this mismatch between what people did and what people say. And that mismatch was highest in the current cocaine users. So the current users, basically less than 50% had that reliability of reporting over their choice, which was explained by the cocaine-related choice. And so if we divide people to those with impaired self-awareness based on that measure, and those with unimpaired self-awareness, so within our cohort, we can divide people, and it actually distributes 50-50% of cocaine-addicted individuals who have that impairment and those that do not have that impairment. They're all addicted. And here we use fMRI bold, and this is response to error on the classical color words true task. And we can see the least activation in the rostral cingulate in those to error, to least response to error in those with impaired self-awareness as compared to the other two groups. And that was associated with cocaine use, so the more the impairment, the more the cocaine use, and with a reduction in the morphological integrity. So this is voxel-based morphometry again, and gray matter in the same region of the rostral cingulate in those with impaired self-awareness. So maybe we identified here a subgroup within addicted individuals with these impairments in self-awareness, awareness, and awareness of the severity of disorder. And maybe as clinicians, we have to think about treating them very differently to the people who have intact self-awareness, because when a person comes to treatment, they're addicted. They can be either aware of the severity of impairment or not as aware, and we want to use different techniques to treat them. So let me summarize. I showed you cognitive impairment and association with the prefrontal cortical integrity and function. I spoke about effects of use, chronicity, abstinence, treatment, attention bias to drug-related cues, and modified neural sensitivity to other salient cues. I did not speak about monetary reward. But there are similar results in alcoholics and association with craving. There are decreases prefrontal activations to other non-drug reinforcers, including erotic stimuli in cocaine-addicted individuals. And that's consistent with the whole concept of the increase of value of the drug reward over a decrease of the value of non-drug related reward, maybe because of adaptations to the supraphysiological stimulation by drugs of abuse. This type of compromised sensitivity to salient stimuli is associated with impairments in decision-making, with depression, anhedonia, with impulsivity. I did mention impaired insight, which we summarized in this paper here. And fMRI may actually detect some differences that can then be predictive of behavior later on. Just to summarize the relapse part of this talk, we see persistent deficits in self-control and reward valuation and addiction. There is impact, of course, of 6 months follow-up in initially treatment-seeking abstinent individuals. We showed a series of studies. So the reductions of drug use over a 6 months period is associated with normalization of both function and structure. It highlights brain plasticity. And beyond providing hope to abstaining drug-addicted individuals, we think that neuroimaging could be useful to track follow-up outcomes. Next, we have to study what are the biomarkers to longitudinally track the treatment course or efficacy of treatment. And I did not mention that, I did mention the concept of abstinence versus current use, but I want you to think about this concept as much more complicated than that. And this is the concept of, for example, incubation of craving and length of abstinence. And here we assess cue-induced activation. This is with the late positive potential I showed you before. you would assume that with abstinence there's going to be a decrease in cocaine craving, but actually it's the opposite. And first there is an increase. Craving incubates, and it incubates the most 1 month after abstinence, but you can see it also at 1 week and at 6 months. And this is the time that this is the time we actually discharge people from treatment. But this is the time they're most sensitive to relapse. So we also can see this type of effects in cognitive function, where the most cognitive impairment characterizes people not who are current users, but those who are abstinent. And that suggests and gives evidence to the self-medication hypothesis, where people use drugs to normalize cognitive function. I'm going to skip that. So some of the targets we can study with meta-analysis approaches. There are some brain regions that are common, and we need to target them. The brain regions do not work in isolation. They are very much connected in a network. And we see evidence, clear evidence for impairments in whole brain type of connectivity impairments in cocaine-addicted individuals. But this is work by Anna Zilberstein that I'm going to present at Human Brain Mapping in Vancouver. I hope to see some of you there. And I want to thank the NAR group, the entire NAR group, and the Brain Imaging Center at Sinai, and your patience and attention. Thank you. Questions? Yeah. So it's the split between what people do, the choices they make on a task, and what they say.
Okay, so you verbally actually.
We ask them what they chose, they tell us, and then we can compare it to what they actually did, and we look at for mismatches. Yeah.
Rita,
Yeah.
wonderful talk.
Thank you.
Question about the methylphenidate experiments.
Yeah.
The effects you're seeing on cognition and connectivity, do you know anything about the pharmacology there? Is that D1, D2, neither, both?
It's the dopamine transporters.
Okay.
Yeah.
But then postsynaptically.
Well, it's like cocaine. So cocaine blocks the dopamine transporter, and
Right.
this one, methylphenidate, also blocks the dopamine transporter, but the time scale is very different. So cocaine peaks very fast in 20 minutes, and this peaks after 90 minutes, and it also stays there longer.
But if you were to hedge your bets on a postsynaptic target, a receptor, is this purely D1, or is this something else?
I imagine it hits dopamine, several receptors, and it's not only dopaminergic, it's definitely also norepinephrine. And that's why we have to compare methylphenidate to other agents, modafinil, but also the ones that target more than norepinephrine in C. And with modafinil, there are also results that are favorable in addiction. But it's work that definitely needs to be done, and we need to look at it more.
Okay,
Thank you.
so much, Rita. Thank you.